First of all, I put forth my sincere thanks to our management of Kalasalingam Group of the institution for accepting wholeheartedly to conduct this 69th International Virtual Learning Series with Chairman Ilayavallal, Dr. K. Sridharan sir. I am here to deliver the welcome address for this inauguration of titled on process and status of vaccine development for COVID-19 in association with the Indian Pharmaceutical Association South Tamil Nadu local branch, head office at Arulmigu Kalasalingam College of Pharmacy. Today we are proud to have an eminent scientist, well-known administrator, and distinguished and honorable research person, Shri Anbu Jaya, Scientific Affairs Director, Retd, Pfizer, Australia, to grace this occasion. We thank you for kindly being the valuable part of this session, sir.
Thank you very much.
Let me acquaint you with the sterling quality of this eminent personality who is a brilliant administrator. Sir, your prestigious presence is a great honor for us and great motivation and inspiration for our entire student community. Sir, we are fully appreciate you for finding the time to be with us in spite of your busy schedule, sir. Most welcome you, sir.
The pleasure is mine.
Most welcome you, sir. Thank you, sir. Next, I want to extend my sincere and special thanks to my dear friend and B.Pharm batchmate, Mr. Jaya from Australia, for his wonderful arrangement of this 69th International Virtual Learning Series. Thank you, Mr. Jaya. Thanks for your wonderful support. It is gratifying to note that this session covers a wide range of interesting topics relating to the process and status of vaccine development for COVID-19. So I hope this session will be a good platform for our students. I trust that every student will put their effort to uplift a successful career. All the very best for all the students. Once again, I welcome you all. Thank you. Thank you, sir.
Thank you. Welcome.
Sir. You can proceed, sir. You continue your session, sir, please.
Okay, thank you. Anbu sir, கொஞ்சம் sharing இது பண்றீங்களா?
Anbu sir.
Sir, sharing தான் இருக்கு சார்.
சரி.
சரி.
இங்க எக்ஸ்ல இருக்கு.
விசிபிள் இருக்கு சார், விசிபிள்ஸ்.
விசிபிள் தான் சார், விசிபிள் தான்.
All right. இப்போ உங்க ஸ்கிரீன் தெரியுதா?
Yes sir, yes sir, yes sir.
ஓகே, சாரி. அனைவருக்கும் வணக்கம். அருள்மிகு கலசலிங்கம் காலேஜ் ஆஃப் ஃபார்மசி and Indian Pharmaceutical Association Tamil Nadu branch. Thank you for organizing this one, and also I take the opportunity to thank the organizing committee, convener, co-conveners, secretary, coordinators, members, and the chief patrons of this college, and the organizing chairman, Professor N. Venkateshan, who just spoke now. I'm happy to be associated with this one because I'm associated with Indian Pharmaceutical Association many years ago. I think I was a treasurer once in Chennai Indian Pharmaceutical Association branch. So it gives me great pleasure to address the students. I always like to be associated with students, even though I'm past that age very long time ago. So what I would like to do today is give you some background and information on the process and status of vaccine development for COVID-19. So I want to split this session into five areas. One is just give you an introduction about basic information about the immune systems in our body, which defends the body from infection. So that's this basic use of the immune system. And the major components of the immune system are skin, mucous membrane, lymph nodes, tonsils, spleen, thymus gland, bone marrow, white blood cells, which includes granulocytes, lymphocytes, mast cells, monocytes, etc. And the appendix. But generally in the common community, they normally think appendix is a waste organ, but it is not so. It is one of the immune system organ. And just to touch about what is the immunity. Immunity is the ability of our body to reject foreign material which includes some chemicals, bacteria, virus, any other organisms, and in particular to resist infection. The immunity basically it's in it has two categories. One is innate immunity, another is adaptive acquired immunity. Innate immunity is the one which we are always born with. That is first line of defense against bacteria and viruses and other foreign substances. Collectively, they are called antigens. The bacteria, virus, and any other chemicals which are not acceptable to our body, they are called antigens. And this mechanism, the innate immunity, is a non-specific defense mechanism. It's not specific towards any antigen. And the response, the negative response against the antigens is very fast. That is immediate response. And this includes physical barriers such as skin, mucous membranes, immune system cells, and acute inflammatory response. As I said earlier, adaptive or acquired immunity. Adaptive immunity, or it's also called acquired immunity, because we acquire over the period of time. So immunity which developed later in life after a microbial infection, especially after a microbial infection. It works against antigens that slip through the first line of defense. As I said, first line of defense is the innate immunity, where the tonsils, skin, everything comes into place. For example, if a bacteria starts coming into our mouth, the tonsils first acts against it, and then it gets inflamed if the power of the bacteria is more. So first we get the tonsillitis and other things. So that's the first line of defense. And when the bacteria passes through that first line of defense without getting affected, then this adaptive immunity takes over. And because it takes over later, it's a slow response, and the antigen needs to be processed by the system first and then recognized by the system. So it takes time. That's why the response is very slow. So once the antigen is processed and recognized by the system, then what happens? Then the adaptive immunity system creates an army of immune cells. The adaptive immune system creates an army of immune cells specific to the particular antigen. We have to remember always the antigen is very specific. The immune cells are specific for the particular antigen. The immune system also remembers the antigen. That's the beauty of this adaptive immunity system. Once we get that antigen, it is exposed to the antigen. Whenever it comes back and attacks us in the second time, third time, whatever, the immune system remembers that one. That's the beauty of adaptive or acquired immunity system. And when I said as earlier, whenever it comes back to us, the antigen, it fights the antigen more effectively when they enter the body next time around. It is more effective than the first time how it was. That's why in the immunity system, they say, if you get the disease in the first time, you may get the disease also when you get the infection. It may be severe in the beginning, but if the same antigen or same bacteria attacks you after a few years, you won't get that much of serious disease. So by the time the immunity system, that's adaptive or acquired immunity, it has acquired the immunity against the particular antigen. or the bacteria or virus. So this is the same principle on which the vaccines work. So coming to the major components of this immune system, mainly the white blood cells or white blood corpuscles. And among that, granulocytes, they promote inflammation and expel large antigens, that is worms and all. And lymphocytes includes B cells, which produce antibodies, which are more important ones. I'll go through that later on. The B cells produce antibodies. The T cells, also called helpers, there are two different major types. One is helper T cell, which stimulates the B cell to make antibodies. If the helper T cell is not there, B cell cannot produce antibodies. That's why it makes the B cells to produce, stimulates the B cells to make antibodies. And the killer T cells, which destroy the infected cells. And another one is a natural killer cells, which the function of it is recognize the virus and kill the virus infected cells. So they all comes under the white blood corpuscles. And also another things which are included are monocytes, which is macrophages. The macrophages is the big ones. They ingest totally and degrade the bacteria and present them to the T cells. And dendritic cells, which antigen presenting cells, they call them antigen presenting cells because they for recognition by lymphocytes. The dendritic cells help the lymphocytes to recognize the antigen. And the lymphatic system, which is is similar to our blood circulatory system. Of course, it travels through the whole body. And the lymph nodes or all the other things are part of the lymphatic system. It carries the antigen to lymphoid tissues to initiate adaptive immune response. And the lymph nodes, it contains millions of the B and C T cells. The B cells and T cells are the most important cells in the immune system. And the lymph nodes percolates the bacteria picked up by the macrophages and the lymphocytes and kill the bacteria or virus. And continue with the major components. Thymus, it produces T cells. Bone marrow produces stem cells, which then comes into WBC, white blood cells, which includes B cells, red blood corpuscles, and platelets. And to easily remember, when you call T cells, it's produced by thymus. And when you call B cells, it's produced by bone marrow. That'll be easier to B for B and T for T. You can remember that. And the spleen. Spleen produces and stores the WBCs and destroys the antigens. That's a function of a spleen. Tonsils, as I said earlier, first line of defense. And mast cells release histamine. Whenever you get some allergic one, the histamine mast cells breaks down and releases the histamine. That's why we get the rash and the things on the skin. That's the purpose of a mast cell. When we talk about the antibodies, what are the antibodies? We know B cells produce the antibodies. They are specialized Y-shaped proteins. Look at you can see on this screen, the diagram, the Y-shaped one is the antibodies. And it stops real pathogens from infecting the cells. Why I call it real pathogen from infecting cells is the parts of the pathogen or the bacteria or virus also is there in in the vaccines. So that doesn't trap that one because that's required to produce the antibodies. So it stops real pathogen from infecting the cells. And the antibodies also called immunoglobulins. And there are 5 categories of immunoglobulins: IgG, IgM, IgE, IgD, and IgA. All the antibodies are classified under one of these groups. And they are, as I said earlier, they are produced by B cells. And like you can see on the diagram, the antibodies, like a lock and key, locks the antigens so that it doesn't attack our cells and trigger a chain of actions to fight the invader. And the antibodies are supposed to be considered as warriors of the immune system. And we have to always remember, any new antigen, there is a new antibody produced in the body. And just as a simple diagram for quick memory, bacteria or virus, we call them antigen. They enter the body and they start multiplying here, bacteria or virus. And at the same time, the immune system is triggered and antibodies are produced by the body or one of the parts of the immune system. And the antibodies enters the circulation and kills the bacteria or virus. And our body got rid of the bacteria and virus and get the immunity. The antibodies stay there, they float in the blood, and we get the immunity. And this immunity is particularly against the particular bacteria or for the particular disease. And we'll go see about vaccines. What are vaccines? Vaccines are medicines which contain an agent that resembles a bacteria or virus and is often made from killed or weakened form of the microorganism or its toxins or one of its surface proteins. I'll show you the surface proteins later on in one of the figures. They help our immune system to work faster and better to protect us from serious diseases. And this is the diagram of the coronavirus. And look at this red arrow, where the red arrow points. It's a spike glycoprotein. This is the part which attaches to the cells of our body and start the infection. So this is the most culprit in the whole coronavirus. And you will see again the membrane protein, nucleoprotein, and the other proteins on this one. We talk about at the later stage. And vaccines may contain a bacteria or virus, parts of the above, or its genetic material. Okay, when the vaccine is containing a virus or a bacteria or parts of them, are they really safe? We really get the doubt, isn't it? Yeah, because they are safe, they are killed microorganisms or weakened microorganisms or undergo, and also before the vaccine comes to the market, for our use, they undergo extensive testing before use. So we are going to see what are the rigorous testings goes for before the vaccine comes into the market. Are there any side effects? Yes, of course, there are side effects. Most of them are minor. And if there is any major, that's why we always say administered under the supervision of a medical specialist or our doctor. And danger due to the infection is worse than the side effects of the vaccine. So in that case, we choose the vaccine. With regard to side effects of corona vaccine, yes, there is no evidence at this stage about what are the side effects because the vaccine is not yet available. Only the vaccine comes out of the market and used by a lot of people, you will understand the side effects of that. At this stage, we are in a black room. We don't know anything about the vaccine, whether there's side effects and all. And as I said about the administered under supervision of medical specialists, some individual people may react differently to vaccines or for that case, any injections, people can react differently. So for that, only when you get a vaccine or any injection, stay in the doctor's place for 10 minutes or 15 minutes, so that if there is any emergency, they will be able to treat you immediately. Instead of taking auto or taking your car and coming back to your room and or to your house and get the problems. So it's better, it's always suggestible, advisable to stay in the doctor's place after you have a vaccine for a while and come to a house, go to our house. Okay, next we see how do the vaccines work? You have seen this type of picture there, similar like how the virus or bacteria works. Vaccine, that's also an antigen now. It enters the body, of course it doesn't multiply here like the bacteria or virus does. And once the vaccine comes, the antigen comes, immune system is alerted and the antibodies are produced. And antibodies, because it's not going to kill anything, any bacteria are there. So it stays in our body and improves the immunity and stays there for life. So if any infection, the same antigen, real antigen comes in, these antibodies will take effect and work against that antigen. So that's the part about how the vaccines generally work. How does the safety of the vaccines assured? And before releasing the vaccine to public use, extensive testing is undertaken. They are called, one is a preclinical evaluation and another one is a clinical evaluation, or they call it clinical trial. And after all this, there is an approval process from regulatory agencies, which I think probably those people who work in the industry, pharmaceutical industry, they know how difficult it is to get an approval from a regulatory agency. If any regulatory agency people are there, bear with me. I'm not blaming you, because the regulatory agency is there to protect the interest of the common public. So the companies, for the sake of getting profit, they may do some wrong things sometimes, and it's the duty of the regulatory agency to protect the public. So that's a big hurdle before any vaccine comes out to the market. So many safety features are there before a vaccine comes into the market. We'll see what is the preclinical evaluation, what's going on in the preclinical evaluation. Preclinical evaluation is always done in animals, not in human beings. Clinical evaluation is in human beings. So what are the details or looked into in preclinical evaluation? Prerequisite to initial clinical trial. Before it goes into the human use, they have to do this one, preclinical evaluation. And it may use in vitro techniques or in vivo techniques in animals, and toxicity studies in animals to assess the potential toxic effects of the vaccine. They will go up to the maximum. What is the maximum level of the dose it can be withstood by the animal? So from that they predict so much of the maximum dose should be like this. If it is very, very minimum, they try to change and the formulation, they change the formulation or the process of making it. Some of the steps they take during this period itself. And range finding studies, that is, what is the range of the dose they can give or the particular chemical they can use. And the preclinical evaluation need to demonstrate vaccine is suitable for testing in humans. Even for testing in humans, they have to demonstrate the preclinical evaluation should demonstrate that or assure you that. And it's aimed at defining the characteristics of the product, indicating the safety and immunogenicity in animal model. It should be safe and it should produce immunity in animal model. And the documenting safety data is always there. I think with the clinical studies as well as the preclinical, the documentation is very, very important, starting from the beginning to the end. And it's a minor incident or major incident, it has to be documented. That's what the regulatory agency will be looking for in any submission. We can't say, oh, this is a minor one, only one person has affected. No, it should be included in the data. And it identifies possible risks to humans, helps to plan protocols for subsequent clinical studies in human subjects. Because before they do anything in these studies, they need to have a protocol prepared, and sometimes they get it approved by the temporary approval from the regulatory agencies. I will explain a little bit later before I start the clinical studies one. And performed in compliance with GLP. I think most of you probably would have heard about GMP, GLP, all those things. GLP is a good laboratory practice and also guidelines for animal experimentation. Because we are doing an animal, we can't do it willy-nilly. So we have to follow certain guidelines for animal experimentation. And clinicians, that is those who are going to do the clinical studies in humans, are responsible to satisfy themselves that the preclinical experiments and the data collected are adequate to proceed to clinical evaluation with regard. Okay. Okay, we have successfully completed the preclinical evaluation, and we got satisfactory outcome in preclinical evaluation. If the answer is yes, move to the clinical evaluation. If the answer is no, you have to abandon the trial and start all over again. Whatever the company invested in this up to this study, it's all waste. It's all a waste. That's why when a medicine comes into the market, the company which is produced it first or released it first would have spent a lot of money on these type of works. That's why the price at the time of release to the market, it is always very high. And sometimes we'll say, oh, this one is only so much price, or 1,000 rupees or 10,000 rupees like that. Yes, because they spend a lot of money and the company has to recover all this money. Next, we'll go to the clinical trial or clinical evaluation. And what are the important points to note in this one? You need to have a sound understanding of the epidemiology of the pathogen. And now we know about the coronavirus, the epidemiology, how the epidemiology is throughout the world. We know, even a common man now knows about it. And proportion of infected people who develop clinical disease. Infection may be they're prevalent, or one person may have been infected with coronavirus, but they may not have a clinical disease. That means the moderate one. They may not have the other symptoms of the coronavirus or Covid19. So those type of people who develop initially a little bit of the antibodies, and that is sufficient to protect them from the infection. And we should know about the risk of transmission from one person to another person about that virus or bacteria. And the evaluation to comply with the WHO guidelines for good clinical practice, GCP. And subjects in the trial, that is the volunteers. And another thing I should tell you, anybody who is involved in this clinical testing or who are subjected to this testing, the company is not supposed to pay anything as a compensation to them. They can only pay to compensate the time and whatever money they lost or for the travel, other things. But monetary benefits is not permitted in these clinical trials. I'll tell you why in the next one or two slides. Subjects in the trial should not be exposed to unreasonable or important risk of illness or injury. Important to ensure that economically or socially deprived communities are not exploited in this research. This is the reason why I said there should not be any monetary benefits for people or volunteers who are joining in the clinical trial tests, because socially deprived communities are not exploited. If the monetary benefits is there, one company can go into a very poor country and pay money and enroll people because they get the money. Poor people may get enrolled and test on them, like making them as a guinea pig. So that's the main principle of this clinical trial rules or the guidelines. We are not supposed to pay anything. And manufacturing consistency in lots need to be demonstrated and well documented. This is, I think it goes without saying. Like sometimes people say, if it goes to a foreign country, the quality of the medicine is very good, and if it is sold in its own country, the quality is bad. To avoid that type of ones, they have this type of conditions. Manufacturing consistency for vaccines lots need to be demonstrated and well documented. This is part of the GMP also, good manufacturing practices. Vaccine lots used should adequately represent the formulation intended for marketing. You can't use one formulation for testing and another different formulation for marketing the product. And the primary endpoints are immunogenicity, safety, and efficacy. Study population need to be large enough to precisely estimate the efficacy. For all vaccine developments, there is a need for a long-term evaluation plan that implies a follow-up period of at least 6 months subsequent to the last vaccination. In this only, because of the pandemic of coronavirus, these timelines are a little bit cut off or reduced based on a little bit of scientific reasoning and all. So FDA also has come down from the strict timelines. I'll explain it a little bit later in the other slides, I think. Okay. Methods proposed for use, study population, that is a test methods as well as the testing methods. Study population, inclusion, exclusion criteria, and other aspects should be clearly defined in the trial protocol. Excuse me. Once again. So everything should be there upfront in the trial protocol. You can't write a protocol and while the study is going on, you can't change it. Okay, it's like a locked in the locker. And the clinical evaluation is generally performed in 4 stages. We'll see what are the 4 stages. Phase 1 trial, phase 2 trial, and phase 3 and phase 4 trial. Phase 1 trial is just small scale studies. Primary focus is determination of clinical tolerance and safety. Defines acceptable safety and reactogenicity, that is expected adverse drug reactions are there, all those things. The reactogenicity includes if for a particular chemical you expect certain adverse drug reactions. They should be present because we are using the same drug or same chemical. And preliminary information on immunogenicity needs to be collected. And dose and method of administration are assessed. This is only assessed like a not 100% confirmed, but it is assessed there. Phase 1 trial. And conducted in research environments with adequate laboratory support. And generally, open label studies. Open label studies is what is the patient getting, patient knows, and the doctor or the administrator, they knows about it. And study subjects approximately 20 to 80 healthy individuals. This figure 20 to 80 is general. It can vary depending on the particular medication or particular chemical or whatever is they are testing. That will be negotiated between the company and the regulatory agency where they are going to register the product. And okay, phase 1 trial, successful completion with the satisfactory outcome in phase 1. If the answer is yes, move to phase 2 trials. If the answer is no, abandon the trial and start all over again. It's similar to the same one I said. And whatever the company invested on up to this end of this phase 1 trial, it's all a waste. They can use probably use the knowledge, of course. And We go to phase 2 trial, and main difference between phase 1 and phase 2 trials is large number of subjects and approximately 200 to 300. As I said earlier, this figure is approximate only now. It can vary depending on the particular medication under test. The test is often randomized trials intended to demonstrate immunogenicity profile of the vaccine in the target population. And phase 2 studies investigate the optimal dose of the vaccine. This is where they do the exact optimal dose. Interval between vaccine doses, whether subsequent vaccine doses is necessary or one dose is enough, those type of things they will determine in this. Same way, number of doses per session or per period. And route of administration, which is the best route of administration, is injection or oral or whatever they decide. Most of the vaccines are injection, except like polio virus and other things. Phase 2 studies also investigate safety profile of the vaccine, duration of immunity, how long the immunity stays, which the body withstands that immunity, need for booster immunization, whether if the immunity goes off in 3 months, whether we need another booster which will help, and if it is helps, how long that helps, those type of things will be studied. Qualitative aspects of the immune response, also they measure that qualitative response. And all these things are in the common one. When we say need for duration of immunity, need for booster immunization, all these rules are almost to be broken in this COVID situation because of the emergency situation. They can't wait too long to determine all these things. Okay, immune response to vaccines, vaccine antigens is carefully evaluated, which is the critical part of phase 2 trials. And before proceeding to phase 3 trial, all these things, the immune response should be evaluated. And according to the FDA's the statistics, it indicates only 33% of phase 2 trials proceed to phase 3 trials. All the 67% goes back to the drawing board. Phase 3 trials. They are large-scale clinical trials. They evaluate the efficacy and safety of the formulation. Vaccine efficacy is the percentage reduction in the incident rate of the disease in vaccinated compared to unvaccinated individuals. And subjects, many thousands, depends on the particular we want to use. Provides expanded safety assessment. Just a quick diagram I found in one of the magazines to say what are the different types of trial design. One set is experimental studies and observational studies. In the observational study, there is no change or intervention is done in the studies. Cohort study, case control study, and cross section. And the experiment is one or more factors are altered and effects examined. That two categories: trials without a control group for comparison. In the control trials, trials with a control group for comparison. Under the control or the compare one, there's a non-randomized in which the subjects allocated to an intervention or control group, but without randomization method. And randomized control, RCTs they call it, randomized control trial. Subjects allocated randomly to an intervention or control group. And the randomized controls are considered the golden standard. Participants, that is, volunteers or participants, an important issue in COVID-19 situation. Generally, phase 3 trials use patients, that is, the individuals or the participant with the particular disease, with the disease in question. In COVID-19 vaccine trial or any vaccine trial, volunteers need to be disease-free to assess the immune response generated by the vaccine, that is the antibody production generally. Or healthy volunteers need to be infected with the virus after vaccination. Whether making a healthy individual with the infecting the healthy individual with a virus, the ethical, generally, we can say it's not ethical, whereas US FDA guidance document, if you look at the date of the documents, release document, one is the 6th May 2020, key criteria for the ethical acceptability of COVID-19 human challenge studies. That's human challenge studies is what we are talking about here. And development and licensure of vaccine to prevent COVID-19, guidance for industry. This is released 2 months ago, June 2020. These two documents are specifically to address the COVID situation because of the enormity of the pandemic. Okay, phase 3 trials. Trial design, randomized, double-blind, controlled trials. So randomized, I explained it there. And what is double-blind? The doctor or the administrator, whoever is conducting the trial, as well as the patient, do not know which. There are two. See, for example, one is a drug, one is a placebo. For example, the sugar tablets, whatever like that we call it. For example, don't know. The patient also don't know whether they're taking the drug or the placebo or the sugar tablet. We can easily to understand that. So that is the, of course, that's considered the gold standard as it minimizes the bias. Suppose if you give a patient, I'm only giving a sugar patient, is he going to be happy? Or is he going to feel, even if he is cleared of the disease, he's not going to feel free. No, I didn't take my medicine, so I'm still sick. That's a bias, they call it. And maximizes the chance that the difference in the incidence of disease is due to true effect of the vaccines. So the randomized double-blind control assures this one. Detection of small differences between vaccine and the comparators or the placebo, we can call it. The RCTs can detect the small differences, whereas non-randomized one detects only large differences are there between these. Okay. The RCT may provide indication of long-term protection and the need for booster vaccination. Duration of follow-up depends on the type of vaccine, disease incidence, immunological marker value, and safety. This is again the pandemic. Our corona pandemic has started a little bit leniently, making the regulators a bit lenient on this issue. Because the follow-up, if they wait for the follow-up, they can't bring the vaccine to the market. And there are concessions or the discussion going on between the FDA and other regulators around the world, how much we can relax. That's a big issue going on. And other trial designs, other than this RCT trial, other designs can be used. Alternative types can also be considered to provide efficacy data, but allowed when they are fully justified. Why we are not using RCT, why we are using the other alternate testing. We have to justify to the regulatory authority. And criteria for the acceptance of the vaccine for use. Clear, definitive evidence that the vaccine is safe. Safety is first. Prevents the infectious disease significantly. Either it prevents the infection significantly or alters the. Now, if you look at the pandemic, history of the disease now, so many people affected, so many people doesn't survive, the survival rate. If it alters this history, see survival is earlier, say for example, only 30% survived. if the vaccine improves the survival rate to 50%, then that is a little bit acceptable in that area. That's what I meant, the alters the history of the disease. Satisfactory completion of phase 3 trials, then it goes to the regulatory submission for marketing approval. And another one is the phase 4 trial, which generally with post-marketing surveillance. This is after you release the product into the market or to the public. Post-marketing surveillance, and most of the big companies, they do this, strictly follow this one. And it describes and quantifies safety profile of the vaccine and determination of long-term protection, determine the need for booster vaccination if there is required, detect rare adverse events. For example, if you are testing only on 3,000 people or 5,000 people in the phase 3 trial, and if there is a reaction which is, adverse event, which is 1 in 10,000, it can happen, 1 in 50, it can happen. You would have heard about this one. Sometimes they say, oh, this reaction is 1 in 10,000 or 1 in 10 or 50,000 like that. That case will be missed out if the study population was only 3,000 or 4,000. So for that sake, they can't do the testing itself for 10,000, 20,000 or 50,000. And so to detect that one, this post-marketing surveillance is very important. And pharmacovigilance activities, regulators a requirement also. Pharmacovigilance is the one which is follows the adverse drug events. And each and every company should have a pharmacovigilance department which perform, or they give it to the licenses to other third-party pharmacovigilance companies. Okay, we talked about the testing profile, testing regimen. Now there's a global search for Covid-19 vaccine. The status as of 22nd August, that is yesterday, the total vaccines in preparation or in trial or going to trial are 160 plus vaccines. And they are in the preclinical stage, about 135 plus vaccines are in preclinical stage. Phase 1 trial, 21 vaccines. Phase 2 trial, 13 vaccines. And phase 1 and phase 2, if they are successful, then go to the phase 3. And phase 3 at the moment is 8. And approved for early or limited use, only one vaccine, which all of you might know now, is in Russia. I'll talk about it a little later. Okay, the categories of vaccine under trial. We can have a broader category of 5, but the 4 are the major ones. The 5th one, repurposed, is the already existing vaccine. The genetic vaccine, viral vector vaccines, protein-based vaccines, whole virus vaccines. There are 4 categories, main categories in this. What are genetic vaccines? Vaccines that use one or more of the coronavirus's own genes to induce immune response. So the one company called Moderna and National Institute of Health testing messenger RNA based vaccine. It protected monkeys from the virus from their preclinical studies. Human trial started in March 2020, launched a phase 3 trial on July 27th, that is last month. And 30,000 healthy people are being tested, are used in the trial, and 89 sites around USA, they are doing it. And they expect to market the product in 2021. And BioNTech and Pfizer, my old company, and Fosun Pharma, testing same messenger RNA based vaccine. Phase 1 and 2 trials caused volunteers to produce antibodies against this virus and T cells that respond to the virus. On July 27th, they announced the launch of phase 2 and phase 3 trials with a total of 30,000 volunteers in the US Argentina, Brazil, and Germany. Expect to file for regulatory review in October 2020. When I talk about this, doing it in different countries, this is more important for generally the new chemicals, new chemical entities, NCE we call it. And sometimes it's mandatory to do different regions because Asian people, they react differently to other country people for certain reasons, like the enzyme differences and all. You might have studied about those things in your studies. That's a bit too detailed one to discuss about that in this area. So I'll go to the next one. Zydus Cadila in India, they started testing the DNA-based vaccine, Zyco-D, they call it, in July. The second company in India to enter the Covid-19 vaccine race. And phase 2 testing launched on August 6, 2020. Another company, CureVac in Germany, testing a messenger RNA, phase 1 in June, and registered for phase 2 in August, to do it in August. Expect to produce hundreds of millions of doses a year. Sorry, I meant yes. Imperial College of London on Morningside, testing an RNA vaccine, registered for phase 1, phase 2 on June 15. Results of the trial expected end of 2020. Another company, Arcturus Therapeutics and Dukeness Medical School. They developed the same messenger RNA-based vaccine, strong immune response in animal experiments they found out, and launched the phase 1, phase 2 trials in Singapore in August this month. And another type of vaccines, viral vector vaccines. What are viral vector vaccines? Vaccines that use a chemically weakened virus to deliver coronavirus genes into cells. The cells make viral proteins to induce immune response, and that immune response is used. So what are the companies? CanSino Bio, Academy of Military Medical Sciences, developed a vaccine based on an adenovirus called AD5, published promising phase 1 results in May. On July 25th, reported phase 2 trials demonstrated the vaccine produced strong immune response. Phase 3 trials to be conducted in Saudi Arabia. And AstraZeneca at Oxford University, this you might have heard a lot about this. And in my opinion, I think they may come out as the first vaccine in the wider market or wider world of the market. Testing a chimpanzee adenovirus called CHASOX1. Study on monkeys found the vaccine provided protection. Phase 1-2 trials in England and India, they did. Phase 3 trials they are going to do doing in Brazil, South Africa, and the US. Manufacturing capacity, they are building or having 2 billion doses manufacturing capacity. For this study, Indian Serum Institute in Pune produced millions of doses for the trials, for using in the trials. And as I said, as I explained earlier, the same method of manufacture and the quality of the product will be exactly the same for what they are using in the trials. Otherwise, they may have to do a bridging studies and all. Johnson & Johnson and Beth Israel Health companies, they joined together, developed a vaccine based on adenovirus 26, launched phase 1 and 2 trials in July 2020. Phase 3 studies in September 2020, they expect, with 60,000 participants, aiming to produce at least 1 billion doses in 2021. And Gamaleya Research Institute, Russia, testing a combination of 2 adenoviruses, AD5 and AD26. It's a combination one. On August 11th, President Vladimir Putin announced that the Russian healthcare regulator has approved the vaccine called Sputnik V before even phase 3 trials had begun. I don't want to comment about this one. Even the phase 3 didn't start, and they have approved it for public use. And Vietnam has agreed to purchase 50 to 150 million doses of this vaccine. This is the only vaccine I said earlier released. Protein-based vaccine. Vaccines that use a coronavirus protein or a protein fragment to induce an immune response. I showed you this picture of the virus, and these are the proteins: membrane protein, nucleoprotein, and of course the spike glycoprotein. They either use a protein or the fragment to induce immune response. This type of vaccines. And Novax or Nucleus Network, Australia. The Novax company is a US company. Nucleus Network is in Australia, testing a vaccine which was made by sticking proteins onto microscopic particles. It's a new method of manufacture. 2 preliminary studies in monkeys and humans provided promising results. August 17th, they launched phase 2 trial in South Africa on 2,900 volunteers. Large phase 3 trial to start in October 2020. Expect to deliver 100 million doses in the US by first quarter of 2021. Anhui Sci-Fi Logcom and Chinese Academy of Medical Sciences testing a vaccine that is a combination of viral proteins and an adjuvant that stimulates the immune response. The adjuvants are added to the vaccines to stimulate to improve the vaccine's ability to induce response. And the famous adjuvant used is aluminum. Alum is used as a generally used for many years. It has been used as an adjuvant to vaccines. And this company launched a phase 2 studies. Vaccine and another Australian company developed a vaccine that combines viral proteins with an adjuvant that stimulates the immune system. Phase 1 completed in July and phase 2 trials in September 2020. Medicago, GSK, and Dynavax uses a species of tobacco to make vaccine. They deliver virus genes into the leaves of tobacco leaves, and the plant cells then create protein shells that mimic viruses. Medicago, Canada-based company, launched phase 1 trials on a plant-based vaccine, that's the above one, in combination with adjuvants from GSK and Dynavax. And phase 2-3 trials to start in October 2020. And University of Queensland, that's in Australia, and Commonwealth Serum Laboratory in Australia. The vaccine is distinct from other candidates as it uses molecular clamp, a technology patented by the university. Their vaccine delivers viral proteins with an adjuvant to induce a stronger immune response. Phase 1 launched in July 2020, hope to produce up to 100 million doses of their vaccine by the end of 2021. University of Pittsburgh developed a vaccine called Pitcovac using a new delivery approach, microneedle array. That the delivery name is microneedle array. It's a finger-sized skin patch. You know, like the smoking to stop smoking, they use the skin patches. Like that finger-sized skin patch, tipped with 400 tiny needles made of sugar. The needles deliver spike protein pieces when placed on the skin. You know, spike protein I talked about in the virus picture, that pieces of that they use. And recruiting volunteers for phase 1 trial. Kentucky Bioprocessing developed a species of tobacco. They developed another species of tobacco, Nicotina benthamia, to make viral proteins. The company used this technique successfully to produce a drug, ZMapp, for Ebola virus. Because they have used the same technique and they are trying to use the same one. Because for the Ebola virus, they did that successfully. So they are using the same method, same type of manufacture or production. Phase 1 trial planned to be launched in July. Next is a whole virus vaccines that use a weakened or inactivated coronavirus to induce an immune response. Wuhan Institute, where the state where the corona pandemic started, Wuhan Institute of Biological Products and Sinopharm, they developed vaccine using inactivated virus. Phase 1, phase 2 trials provided promising results. Launched phase 3 trials in the United Arab Emirates in July 2020 and in Peru and Morocco in August 2020. Sorry. Beijing Institute of Biological Products, Sinopharm, again, developed a vaccine using inactivated virus and launched phase 3 trials in the United Arab Emirates in July 2020 on 5,000 volunteers of this version and another 5,000 volunteers on the Wuhan Institute version of the vaccine. Sinovac Biotech is a China company again, testing an inactivated virus vaccine called CoronaVac, launched phase 1, phase 2 trials in June on 743 volunteers, launched phase 3 trials in Brazil in July 2020, and in Indonesia they started in August 2020, building a facility to manufacture up to 100 million doses annually. Next is our Indian company, Bharat Biotech, ICMR, and National Institute of Virology designed Covaxin, a vaccine based on an inactivated form of the coronavirus. Phase 1-2 trials started in July 2020. There was a false reporting in the papers and also by the ICMR director. Vaccine will be ready by August 15th, Indian Independence Day. You know the joke about that one. It is likely to be available for public use before the end of 2020 or early 2021. In this one, they are in a hurry. They announced that it will be released on August 15th because it was Independence Day. And they didn't realize how much of steps that a vaccine has to go through before it goes to the public. And the last one is the repurposed vaccine. The vaccines already in use for other diseases that may also protect against Covid19. They are testing about this one. And Murdoch Children's Research Institute, Australia, they are conducting phase 3 trials because they don't have to go through phase 1, phase 2 because it's already an existing vaccine. They directly go into the phase 3 trials. Using the TB vaccine, BCG vaccine against coronavirus, determine whether the vaccine partly protects against coronavirus. And some projects, MGR University, Chennai, they were working on a vaccine and no further information available. They announced earlier they are working on a vaccine. Dr. Sudha, I forgot her name. She is the chief of that MGR hospital, University Hospital. And Japan, Thailand, Australia, all these countries also working on vaccines. And our expectation, as we have seen, all these trials are going into phase 3. Next phase 3 is marketing approval and getting into the public. Hopefully, successful completion of phase 3 trials will result in marketing approvals from the regulators, and the vaccines for COVID-19 is available for public use. Let's hope for the best. And these are the references. If you want, anybody can copy it when it is there. And more details are there in these references. With that, I complete my presentation. Thank you very much. I take this opportunity to thank Arulmigu Kalasalingam College of Pharmacy, Indian Pharmaceutical Association Tamil Nadu branch, and the faculty members of this college, and the chief patrons, and the organizing committee chairman, Professor Dr. Venkateshan, and our common friend, Mr. Jaya, who is living here. He listened to my talk in Tamil in the local radio, in the national radio here. That was meant for only the public, which is a very basic level, and it's a common language I used in that one. And Dr. Venkateshan heard that one and invited me to provide some more information for your college. So I agreed to that. Hopefully I delivered what he expected. And I take this opportunity to thank each and every one of you who had listened to this one. Thank you very much. If any questions, yeah, you can shoot.
Thank you, sir. Thank you, sir. Really, we had a informative and elaborated, nice presentation, sir.
Thank you very much.
I hope this topic will motivate the students and encourage them in active learning in forthcoming days.
Yeah, hopefully. Yeah.
Yes. Any questions?
Thank you, sir. So there is a question in the chat box from Mr. Raghavendran, third from D. So during the medical emergency, it is necessary to test it on animals.
Yeah.
Why we won't go for given human test directly?
Now, that's what I said. The safety has to be evaluated. And you know, maybe in our country, maybe it's a different one. But if any company, without doing this process testing and test on human beings, if one person gets affected, you know, the company may be paying compensation. They have to pay compensation for losing a life, or if somebody gets affected by that vaccine, it's a very dangerous situation. That's why I think another thing I forgot to mention, even yesterday, I think Donald Trump, our president, Donald Trump accused FDA of purposely delaying the approval of this process, the vaccine manufacturing process, which is, in my opinion, is ridiculous, because politicians, they have to stay in where they have to. This is not a medical issue where he can talk about. Earlier, he talked about the hydroxychloroquine chloroquine tablets, and now the US FDA banned it, or it's not being used in America. So this is the decision to be left to the technical experts, the medical experts. And you can't go into human testing immediately. Nobody will be accepting it, because even to recruit the volunteers, it's very difficult. So it's not ethical. That's why.
Okay, sir. Thank you, sir.
Yeah.
So there is another question from Dr. Jaya.
Yep, sure.
How long it takes to release a vaccine? What will be the minimum possible period to release a vaccine?
I think generally it's 18 months to 24 months. With everything goes on well, and that's the normal time limit. The minimum one they will do it, but they are trying to reduce it a little bit. That's why I showed you the two documents, the June 2020 document from FDA. If you go and read that one, they give you a little bit of relaxation where they can relax. They can't relax totally. As the other gentleman asked, why don't you go directly to human testing? They can't do that, because as a health regulators, they have the responsibility to the life of individuals. They are the only people who can protect the common man or common individual. So they can't willy-nilly take the decision. And there are set procedures which have been very useful, and it has been found successful. So they follow it.
Thank you, sir. There is another question from Mr. Saugul Hamid from Third B Farm.
Mm-hmm.
Instead of going to human trials, there is any other possibility to test the vaccine?
No, not in the virus one, because they do the animal testing already. and animal testing is successful. They have to do it because animals, any medicine or any chemical behaves slightly different in animals and slightly different in human beings. Sometimes totally different also. One can be acceptable to the animal, and that cannot be totally acceptable to human beings. That's why in the phase 1 trial, they only use few people. I said 20 to 30, 20 to 80 people, because they don't want to go into larger population and make everybody suffer. That's why step by step they go. Phase 1, phase 2, phase 3.
Thank you, sir.
Yes, Carl.
Sir, since you have been in Australia,
Yeah.
I came to know that that immunization practices are not that much practiced over there in Australia like India.
Sorry,
It was
sir.
true, sir.
Can you
No,
repeat the
sir.
question? I missed the words.
Immunization schedule for infants. My cousin is there in Australia.
Right.
She told here we are not having that much immunization schedule as like in India. It was true, sir. If it was true, how the vaccine is going to be prevent the COVID, sir? Like in developing countries, they are not having much practice of immunization like developing countries.
Immunization.
Yeah, immunization, vaccines.
Oh, generally immunization here, if somebody gives birth, the doctor itself recommends and gives a good, like a booklet they will give. This is the time, this is the vaccine, when you have to give, when you have they have to take that to the general practitioner. It's general practitioner itself. So from the time the child is born here, they are taken care of. But there are certain set of people who don't want to have vaccines given to their children. That's their option. They don't force. Especially some religious groups, they don't want to have any medicines also, for example. Is that answer your question? I didn't
Yeah,
really.
yes, sir. Yes, sir. Yes, sir. She also told the same thing. Here, the immunization schedule is not mandatory. It's a.
No, I won't say mandatory. I don't say mandatory. It is available and it's streamlined here. But if somebody doesn't want to, they don't force you. Because this country is a free country, so you have the freedom as long as the freedom doesn't affect the other person.
Okay, sir. Okay, thank you, sir.
Welcome.
So, based on a scientific report that COVID-19 virus is mutated frequently, how the vaccine is going to be prevent the spread of disease? Because it is mutated, there may be a change in the antibody.
Yeah. That's true. I think that's a good question. The mutation, they can't expect. I mean, nobody can predict that one how it goes. We have to take it things as it stands today, because it's getting mutated. We can't keep quiet. Oh, let it finish the job, then we will do it. No, we have to do something like that at this stage. So that's the steps in evaluation. If that comes, then that time we have to fix it.
Thank you, sir.
Welcome.
Sir, there is another question. Like recently, there is a mutation called Suanga cluster in Malaysia.
Yeah.
Which will spread more faster at a higher rate.
Yeah, I read that.
Actually, it was said to the same thing, sir. Like if it is like mutating very fastly.
Yeah.
And when the vaccine was coming into force, and they are going to be give it a shot or give it a try.
Yeah.
So if it won't work, what we can do, sir?
Yeah, that's true. I think sometimes we have to work on the current scientific evidence. That's all. I think what nobody predicted the coronavirus is not a new virus. It has been there, and this is a new form, little bit of new form, which has affected so much. Nobody expected this; it will affect this much as become as a pandemic.
Thank you, sir. And there is a conspiracy like it's a human-made disaster like that. So your opinion on this?
No, I won't. I don't want. I won't comment on that because it's all. Sometimes people say it's maybe a rumor. There may be some fact. I mean, I don't want to be like American president to jump into conclusion to blame some other country. Sorry.
Thank you, sir.
Okay.
So, who is going to be released the vaccine first, sir, according to you? Because Russia already announced they are on human trial. Like India also they had told on August 15 and they told another 3 months it will be ready like that. There are a lot of theories are going over there.
Yeah.
So based on opinion from you,
I think the AstraZeneca one is the likely candidate, I would think, because they have come very far and they are doing everything. And in India, they may easily introduce because they already have the infrastructure there with the Pune Institute. And they have come a long way. I think probably AstraZeneca's product will be the first to the market. And the Russia one, I don't want to talk about that because if they don't want to follow the scientific ones, there's no point in talking about that. And they are using the whole population for the phase 3 trial. That's what I would say. They didn't go for the phase 3 trial, and they are using the common man, all the population as a phase 3 trial. Do you think it is right?
maybe, sir.
Yeah. That's a bit. I'm happy I'm not a Russian now.
Oh, okay, sir. Okay. Thank you, sir. Any other questions from students? Any other questions?
Okay, Anbu. Thank you, Anbu. Thank you, Anbu. Thank you. Hello.
Yes. Solunga.
Thank you, sir. Thank you. Sir, on behalf of our management of Arulmigu Kalasthalinga College of Pharmacy, hereby I express my wholehearted thanks to you and your thank you. Sorry, sir. Thanks to you for giving such a informative and elaborated presentation and interaction also about the. Can you hear, sir? Can you hear?
Yeah, I can hear you. Thank you very much.
Yeah. Process and strategy. Can you hear, sir?
Yeah, I can hear you.
Yes, sir. About the process and status of vaccine development for COVID-19. So some seed of intelligence in our students. In future also, sir, let's have a good collaboration for the development.
Sure, anytime.
Thank you, sir. Thank you, sir. Whenever would like to plan to Tamil Nadu, sir.
Yeah.
In future, sir, please let me know. I will invite you in our institution, sir. Please, sir.
Sure. I'll do that. Yeah. I used to visit Madurai often because of the corona only. I stopped now.
Yes, sir. Yes, sir.
I was there in February.
February coming 21st.
No, last February I was there. Just before corona started.
Before started. Okay, sir. Please, sir, inform, sir. Next time,
sure,
please inform.
Definitely.
Know, sir. We will invite our institution, sir.
Sure, yeah.
Sir, another one more small request, sir.
Yeah, sure.
Sir, kindly let us know, sir. Kindly refer, sir. Any of your friends who are able to guide help in the development of our pharmacy students, sir, for webinars, sir. Particularly in webinars, international webinars. Kindly refer, sir.
Yeah, sure.
Refer your friends or colleagues.
Yeah, definitely. I'll get in touch and check with my friends also.
Thank you, sir. Thank you, sir. Very kind of you, sir.
Thank you.
Yes, sir.
Thanks for the opportunity. Thanks for the opportunity. I thank all the students who patiently listened to it, because the first 1/3 of the presentation was purely the classroom type of subject.
Yes, sir.
So bear with that. So without that, I think they can't understand the phase 1, phase 2, and why the arguments and all they can't understand. So I thought I'd give the basic information.
Basic information. Thank you, sir. Thank you, sir.
Okay, welcome. Thank you very much.
Take care, sir. Take care, sir. Bye.
Take care. Have a good night.
Good. Bye.
Thanks to Anbu sir also. Yes.
Thank you, sir. Thank you, sir. Thank you.
Thank you. Good night. Thank you.